Good afternoon, dear friends, and welcome to this session of our cluster complex meetings. So, in this session, we will discuss some critical and difficult concepts found in our APSCRT 10th class biology textbook from chapter Life Process to Reproduction, which covers the syllabus for FA2. Though the level of understanding differ from school to school, teacher to teacher, and student to student, on the whole, we can generalize some difficult concepts. Difficult concepts in the class 10th biology textbook often includes advanced details in life processes like digestion, circulation, excretion, control and coordination due to their complex mechanisms. A lot of difficult and critical concept should be redressed and discussed in this session. So due to lack of time, I would try to discuss five most important critical concepts in this session. So they are on the slide. That is one from human digestive system, structure of the heart and its function, difference between blood and lymph, division of human brain and their components, DNA copying and variations, and much more. So here in the first chapter itself, whenever the human digestive system is concerned, so far it is concerned, we both the teachers and students feel a little bit challenging in memorizing the parts involved in the digestive system and their function. So we can use this type of chart for a better understanding of the digestive system, human digestive system, because we know that the digestion is nothing but the breaking down of larger complex food materials into simpler complex food material. That is, the carbohydrate is digested into glucose, proteins are digested into amino acids, and fats are digested into fatty acid and glycerol. So this type of flow chart supporting with a diagram and the concepts mentioned in the boxes like a flow chart will be helpful. You can see that the digestive system starts from mouth itself, and also the salivary glands, and it turns to pharynx, esophagus, liver, stomach, small intestine, pancreas, large intestine, appendix, and anus and rectum. So we can easily understand if we use this type of flow chart just mentioned in the slide and their function. So concisely I will discuss, because this type of chart, when we displayed in the classroom in the IFE boards, the children can easily grasp the parts of the digestive system and their function. For example, we can start with the mouth is the initial point of ingestion, and after that the mouth has dentitions and dental teeth organization that helps in the breaks of the food particles, and the salivary glands present in the mouth and the buccal cavity moistens and lubricates food first, and an enzyme called salivary amylase or ptyalin which cleaves the carbohydrate or starch. Then the food enters into the pharynx. After swallowing, it enters into the esophagus with a movement called peristaltic movement, which transports the food into stomach. So stomach is a bag-like structure. It helps in churning the food, just like a grinder is used in our kitchen. How the grinder itself churns different food materials and makes it a semi-solid material called chyme. And here a acid called hydrochloric acid present in it, which activates enzymes like pepsin and helps in the breaks of the food, and it also kills the germs. And a layer of mucus protect the stomach wall, and after activation of the pepsin, it cleaves the protein. So there is a limited absorption of food takes place in the stomach, and the stomach has also some association glands besides them, namely the largest gland of a human body called liver, what you can see in the flow chart, which breaks down and builds up many biological molecules. It also stores vitamins and iron, destroys old blood cells, destroys poisons, and the main and the prime function of the liver is production of bile, which helps in the emulsification of the fats, and it also stores the bile. And there is a bladder present underneath the liver, which is called gallbladder. And beside the liver, there is also a mixed gland. There is a gland called mixed gland, pancreas, which has the function of exocrine glands and also endocrine glands, as a leaf-like structure present underneath the stomach, and it regulates blood glucose levels by secreting a hormone called insulin, which is also we can learn in the control and coordination system lesson. And here the bicarbonates are neutralized by stomach acid, whereas trypsin and chymotrypsin are otherwise called proteases cleaves the proteins here, and the carboxypeptidase cleaves proteins, and amylase cleaves starch and glycogen. And also pancreas secrete a lipase, which also cleaves lipids and what is called emulsification and saponification of fats. And nucleases also cleaves the nucleic acid, which is not mentioned in our textbook. They will learn in the further studies. And now the food material enters into small intestine, where the completion of digestion takes place. Inside the inner layer of the small intestine, mucus protects the gut wall, and it is the absorption of complete nutrients into the blood takes place through villi, and the proteases cleaves proteins here, sucrases cleaves sugars, amylases cleaves starch and glycogen, and also bile aids in the digestion, and lipase cleaves lipids. So here almost all digestion process has been completed and also absorbed into the blood through the finger-like projections called villi. Now the indigested food enters into large intestine, where reabsorption of water, ions and vitamins takes place, and it is the site of storing waste materials or semi-digested foods or indigested food, and this is the main part of absorbing water, iron and vitamins again into the bloodstream. Now the blood enters into the rectum, where the indigested food in the form of stool or fish is stored there, and while the rectum is more than half filled, and a stimuli generates from the brain, and the excretion takes place in the form of defecation through the opening called anus. So in this way, by using this type of flow chart with help of diagram, this concept can be made clear to the students easily. So where the problem comes here, what are the digestive enzymes, where they are produced, where they are released in, what is their pH level and how they are changed. So we know that most probably we take major nutrients called carbohydrate, protein and fat digestion, and the enzymes involved in digesting carbohydrate, protein and fat is displayed on the board, and their pH nature is asked in the examination point of view. We have added the pH level of the different enzymes. As I explained in the last chart, how the food are converted into what stage are their intermediate stages. Now in this chart we have not discussed, and in the diagram also we have shown the location of the different glands and their enzymes. So by using this type of chart and a table form, we can easily make the students understand the concepts and make them clear and in a simpler way. So now coming to the point, the second, the most difficult concept which the students feel a little bit difficult is internal structure of heart and the circulation of blood throughout the body. We can use this type of chart to give them a simpler idea, and also we will take help of digital tools like YouTube videos or the videos made by different teachers throughout Andhra Pradesh. So in this slide I am going to explain you first about the internal structure of the heart. So you can see the structure of the heart. The heart is a cone-shaped muscular pumping organ present in the thoracic cavity of human body or chest cavity of human body, a little bit tilted towards left. And the heart is mainly divided into 4 chambers, that right atrium, right ventricle and left ventricle, and those chambers are divided through a thick walls called septa. The prominent character of flowing of the blood is the presence of valves. So we have 4 valves in the human heart, namely bicuspid valve or mitral valve or tricuspid valve, and aortic valve and pulmonary valve. So you see, and the heart is attached with large pipes called vena cava and aorta. So the upper part, the heart collects deoxygenated blood from the upper part of the body with help of a superior vena cava, and if inferior vena cava helps in collecting deoxygenated from the lower part of the body, and the oxygenated blood is circulated throughout the body with the help of a little bit thinner blood vessels than that of the veins called aorta or arteries. So now the heart is now the heart pumps the blood with a contraction and relaxation of the cardiac muscles called systole and diastole. So when we discuss the point coming to the flow chart, the circulation of blood, so the deoxygenated blood is collected from the body by the superior vena cava and inferior vena cava and enters into right atrium. So the right atrium is filled with deoxygenated blood, and when it contracts, it enters the blood into the right ventricle through tricuspid valve. And there is no passage to go on through outside the heart. So whenever the right ventricle contracts or the systole of right ventricle takes place, it enters into the pulmonary artery through a valve called bicuspid or mitral valve. Then the blood enters into the pulmonary artery and then it enters into the lungs. So here the points to ponder are the critical point that the blood in the right atrium is deoxygenated blood, and the right ventricle is also deoxygenated blood or a blood rich with carbon dioxide, and the blood present in pulmonary artery is also deoxygenated blood, whereas the other arteries distribute oxygenated blood, but this is the special type of artery where deoxygenated blood is taken to the lungs for the purification or removing out carbon dioxide and adding of oxygen in the alveoli of the lungs. Then again, from the pulmonary artery, the blood enters into lungs and then again it enters in the left atrium. So here the point to remember that the left atrium has oxygenated or oxygen rich blood. So whenever the left atrium contracts or systole takes place, it enters into the left ventricle, and after the left ventricle it.
Your heart is a pump. It's a muscular organ about the size of your fist and is located slightly left of center in your chest. Your heart is divided into the right and left side. The division protects oxygen-rich blood from mixing with oxygen-poor blood. Together, your heart and blood vessels comprise your cardiovascular system, which circulates blood and oxygen around your body. In fact, your heart pumps about 5 quarts of blood every minute, and it beats about 100,000 times in one day. That's about 35 million times in a year. Oxygen-poor blood, blue blood, returns to the heart after circulating through your body. The right side of the heart, composed of the right atrium and ventricle, collects and pumps the blood to the lungs through the pulmonary arteries. The lungs refresh the blood with a new supply of oxygen, making it turn red. Oxygen-rich blood, red blood, then enters the left side of the heart, composed of the left atrium and ventricle, and is pumped through the aorta to the body to supply tissues with oxygen. 4 valves within your heart keep your blood moving the right way. The tricuspid, mitral, pulmonary, and aortic valves work like gates on a fence. They open only one way and only when pushed on. Each valve opens and closes once per heartbeat, or about once every second. A beating heart contracts and relaxes. Contraction is called systole, and relaxing is called diastole. During systole, your ventricles contract, forcing blood into the vessels going to your lungs and body, much like ketchup being forced out of a squeeze bottle. The right ventricle contracts a little bit before the left ventricle does. Your ventricles then relax during diastole and are filled with blood coming from the upper chambers, the left and right atria. Then the cycle starts over again. Your heart is nourished by blood too. Blood vessels called coronary arteries extend over the surface of your heart and branch into smaller capillaries. Here you can see just the network of blood vessels that feed your heart with oxygen-rich blood. Your heart also has electrical wiring, which keeps it beating. Electrical impulses begin high in the right atrium and travel through specialized pathways to the ventricles, delivering the signal to pump. The conduction system keeps your heart beating in a coordinated and normal rhythm, which in turn keeps blood circulating. The continuous exchange of oxygen-rich blood with oxygen-poor blood is what keeps you alive.
The pathway of blood flows through the heart. Oxygen-poor blood from your body returns to the right atrium of the heart. Blood from your upper body returns through the superior vena cava. Blood from the lower body returns through the inferior vena cava. As the right atrium is filled with blood, it contracts, the tricuspid valve opens, and blood is pumped into the right ventricle of your heart. When the right ventricle is full, the tricuspid valve closes to prevent blood from flowing back into the atrium. The right ventricle contracts, pulmonary valve opens, and blood is pumped into the pulmonary artery and to your lungs. The pulmonary valve closes to prevent blood from flowing back into the ventricle. Oxygen-rich blood from the lungs returns to the left atrium of the heart. As the left atrium is filled with blood, it contracts, the mitral valve opens, and blood is pumped into the left ventricle of your heart. This occurs at the same time as the right atrium pumps blood into the right ventricle on the other side of the heart. As the left ventricle is full, the mitral valve closes, the aortic valve opens, the left ventricle contracts, and oxygen-rich blood is pumped into the aorta to reach all parts of your body. This happens at the same time as the right ventricle pumps blood into the pulmonary artery on the other side of the heart. The aortic valve quickly closes to prevent blood from flowing back to the heart. Meanwhile, the atria have filled with blood and the cycle repeats itself.
So we will ponder another critical concept from the transport system. The main concept here, the difference between blood and lymph. You see, whenever the blood circulates throughout the body, the blood contains RBC, plasma, and nutrients, vitamins, and some fluids, which is in the state of a connective tissue or otherwise called fluid connective tissue throughout the blood circulatory system in veins and arteries. So, where the veins and arteries are made up of semi-permeable membranes, whenever the pressure exerts in the form of systolic pressure and diastolic pressure, some liquid passes through the circulatory system into the intercellular spaces of our body in the form of a clear liquid, which is also called tissue fluid. Those liquids passing from the tiny pores of the circulatory system, veins or arteries, has been collected and stored in the interstitial places or intercellular places, which is called lymph. And lymph is a colorless protein poor fluid because it lacks protein that forms from tissue fluid. It lacks red blood cells and platelets, and they are circulated. Those lymph, wherever the lymphs are collected in the intercellular spaces in our body, they should be distributed and throughout the body. And there is a particularly another type of or a special type of vessel system is also present in our body apart from circular veins, arteries and capillaries, and those are called lymph vessels and lymph nodes. That is both lymph nodes and lymph vessels together called lymphatic system. And this lymphatic system primarily important for the immune defense and drainage because it contains lymphocytes are the microscopic policemen of our body, the defense system, the main organ of the defense system. So whenever we think of the blood and lymph, so there is a distinct difference in between blood and lymph because blood is reddish in color while lymph is pale yellow in color. So red blood cells are abundantly present in blood, whereas red blood cells are totally absent in the lymph. So in other way, we can say that blood minus RBC is lymph. And blood is circulated in arteries and veins, though it has two directions. So away from the heart and towards heart, that is what we call, it is a bidirectional flow, whereas the lymph has to only one direction and it has go to the, again to the heart with the help of some clavillary veins or clavicillary veins. So this is a unidirectional flow. So in the circulatory system, blood flows rapidly. And we know the pressure of the blood is 120 by 80 millimeter Hg. But here in lymphatic system, the flow of lymph is very slow. In the blood, the leukocyte count relatively lesser than that of the limbs, whereas lymph contains high leukocyte count. We know that again I am to remember that leukocyte is nothing but white blood corpuscles. Platelets are present in the blood where platelets are absent in the lymph. So primarily this is the thing. So this is the clear concept we can make with the help of this type of table to the students to make the understanding in between blood and lymph. So here let us together try to simplify another concept where the people feel very difficult to understand, a little bit confusion about the division of brain from the chapter control and coordination. I have to remind you that there are no hard, fast, or strict rules in division of the brain in the anatomy. Still, the scientists or the biologists have divided the brain into three different parts as shown in our figure and also the slide. The major divisions of the brain has been divided into three parts: cerebrum, cerebellum and brainstem. We can see the cerebrum is the largest part of the brain, which is responsible for higher functions such as thinking, memory, emotions and language transformation and transcription. And it is divided into again right and left hemispheres and is covered in folds called gyri and the valleys called sulci, which we will discuss in the next slide. And the middle part is called cerebellum, which is also located in the back of the brain. You can see a pumpkin-like structure with pons and medulla and cerebellum. The cerebellum you can see just a flower-like or a pumpkin-like structure located underneath the brain, which is helpful and crucial for coordinating movement, learning new bicycle and bike management and postures and balance. So there is another level called brain stem, which is situated in the base of the brain, connecting into the spinal cord. So this is the bridge in between brain and spinal cord, which controls essential automatic functions including breathing, heart rate and sleep-wake cycles. So this is the most important organ in the brain nervous system of our body. So whenever we see the next slide, again there are so many subdivisions that is called forebrain, which is the and midbrain and also the hindbrain. So the forebrain includes the cerebrum and its parts like thalamus and hypothalamus, which handles sensory information from different sensory organs, and also automatic nervous system or ANS, and cognitive process, which is linked with the knowledge and understanding, especially the conceptual understanding of the human being has been carried out by forebrain. So the midbrain, which is located between the forebrain and the hindbrain, it plays a role in the processing sensory information and motor responses. Simply, this is just like a CPU of our computers. So the role of the central processing unit can be compared into the midbrain. So the last part of the subdivision is hindbrain, which includes the cerebellum, pons and medulla oblongata. The pons and medulla are part of the brain stem, what we discussed in the earlier slide, controlling vital functions and relaying the signals between the brain and the spinal cord. So biologists also proved that whenever brain has been damaged, there is only a unconscious state. But whenever the spinal cord is highly injured, then it leads to the death. So we can see again in this the diagram how the cerebrum is divided into different lobes. So you see each cerebral hemisphere is divided into four lobes. We discussed in the earlier slide that there are two hemispheres called right hemisphere and left hemisphere, which is also shown in the reddish and a little bit brownish in the slide. And the hemisphere is divided into four lobes. The first one is the frontal lobe, which is pinkish color, what has been displayed on the screen. It involves in the complex cognitive functions, just like learning new things, remembering new things, keeping memory of the new things, synthesizing things, analyzing the things, making connections, understanding the concepts easily, and all the cognitive functions are and planning and the personality is controlled by the frontal lobe of the hemisphere. And whereas the parietal lobe, which is a little greenish, pale greenish color displayed on the screen, processes sensory informations from the body such as touch, pain, and temperature. Now, the third lobe called temporal lobe, which is also a pale color seen in the displayed on the screen, responsible for processing auditory information, memory, and recognizing different stimuli like pinching and crying and all the emotional, internal emotional and external emotional stimuli are controlled by the temporal lobe of our human brain present on the lobes. Now the last one is occipital lobe, primarily processes the visual information. So both temporally lobe and occipitally lobe together processes audio visual synthesis of the things around us, their changes and their analyzing and synthesis process of the auditory and visual are taken place by the temporal lobe and occipital lobe, which is displayed on the screen, and in this way we can make the students learn easily the major divisions of the brain. So now coming to the point, the most important thing in the reproduction system is mitosis and meiosis, what we learned in the 9th class. So whenever the mitosis takes place, children are clear that the chromosomes present in the parent cell is equal to the number of chromosomes present in the daughter cell. So if we take an example, if 44 chromosomes are present in a nucleus of a certain human cell, whenever the cell divides into two daughter cells, each cells contain again 44 number of chromosomes. So there the confusion begins that whenever the parent cell contains 44, how does the daughter cells, when divided into two identical cells, how does the nucleus again contain the same number of chromosomes? So this is made possible by the copying of DNA in the synthesis phase of the mitosis. So the copying of DNA is otherwise called a replication. So the replica of the chromosomes are taken place, whereas the parent strand of DNA or deoxyribonucleic acid, the two daughter strands, just like a Y-shaped fork, is formed at the time of replication of the or copying of the DNA, what we can easily understand with the help of the next video, what I am going to show you. In the process of DNA copying, a parent cell creates additional copies of its DNA through a complex series of chemical reactions. Mainly, there are two types of enzymes involved in it. So, in this video, we can understand, and there are enzymes like helicase and DNA polymerase. One is called scissor and one is called additive or adhesive enzymes present. Whenever the one of the DNA, which is in the double helical structures, are just like a circular loop, is cut along the longitudinally with the help of a enzyme called helicase, where the DNA unwinds and changes into two identical DNA templates. After that, the DNA polymerase adds complementary nucleotides called adenine, thymine, cytosine and guanine to create two new double-stranded DNA molecule, each containing one original and one new strand, what can be understood easily with the help of this YouTube video. And this ensures that when a cell divides, both daughter cells receive the exact same genetic information as the parent cell. So let's watch this video, and easily you can.
Let's take a look at DNA replication, the process in which DNA copies itself. Why does DNA need to copy itself? Well, before a cell divides during mitosis, it must make a copy of its original DNA. This ensures that both resulting daughter cells will have DNA that is identical to the original cell's DNA. So, at what point in the cell cycle does DNA replication happen? DNA is copied or replicated during the S phase of the cell cycle. A good way to remember this is that S stands for synthesis, which means to make. Now, let's see how DNA replicates during the S phase. If we zoom into the nucleus, we can see the DNA molecule's two strands or sides twisted together in the classic double helix formation. The two strands are connected by hydrogen bonds between their nitrogenous bases. The first step of replication begins with an enzyme called DNA helicase. The suffix ase or ACE tells you right away that helicase is an enzyme. Helicase unwinds and then separates the two sides of the DNA molecule by breaking the hydrogen bonds between their nitrogenous bases. Each separate half of the DNA can now serve as a template or pattern for the creation of a new strand of DNA. This separation exposes all of the nitrogenous bases on both sides of the DNA to the environment within the nucleus, where free nucleotides are present. This brings us to the second step in DNA replication, the creation of two new identical DNA strands. An enzyme that assists in this process is called DNA polymerase. It adds free nucleotides available in the nucleus to the original template strands. DNA polymerase does this by creating new hydrogen bonds between the available nitrogenous bases of the free nucleotides and those on both sides of the original DNA molecule. New nucleotides are added to the template strands following the base pair rule of nitrogenous bases. Remember, in DNA, adenine always bonds with thymine, thymine always bonds with adenine, guanine always bonds with cytosine, and cytosine always bonds with guanine. So, these newly attached nucleotides form a mirror image or complementary strand on each template strand of the original DNA. As a result of this replication process, two duplicate molecules of DNA are produced from the original DNA molecule. With DNA replication now completed, the cell is ready to begin mitosis. We will cover mitosis in another video. In summary, DNA replication is a process in which DNA duplicates itself, making an identical copy. Replication occurs during the S phase of the cell cycle when a somatic cell is preparing to divide. DNA replication is necessary so that the two daughter cells produced after mitosis both have DNA that is identical to each other and identical to the DNA in the original cell. At the beginning of replication, an enzyme called DNA helicase breaks the hydrogen bonds between nitrogenous bases and unwinds the DNA molecule. Each side of the original DNA molecule serves as a template for the creation of a new complementary strand of DNA. With the assistance of DNA polymerase and other enzymes, free nucleotides are added to the template strands following the base pair rule. The result of DNA replication is two identical duplicate DNA molecules from the original DNA molecule.
Watching the video, again we are here to discuss about the importance of DNA copying in reproduction. So why should DNA replication should take place? Because it contains information about inheritance from parents to offsprings, and it codes for proteins required by an individual. If DNA is not copied during reproduction, the correct proteins will not be formed, and the structure of the individual will be entirely different. And we know that sometimes whenever the errors in DNA copying happens, that leads to emerging of new characters which is not found in the parent characters. So some new traits are expressed because of the errors in DNA, and that leads to the evolution.